Thank you very much, Joe, and everyone at OcuMel UK for inviting me. Yes, Sarah couldn't be here. She's incredibly busy, but she sent me instead. So my name's Helen Kalirai. My first disclaimer is I'm a scientist. I'm not a clinician. And so please be gentle with me because, yeah, it's very different. I've really enjoyed all the discussions, all the patient discussions. It's very informative for us. And I think it's something that we encourage all our students and everyone that we have working in the labs to sort of get involved in, patient and public involvement. And so I've got a fancy title that Sarah gave me, but essentially, as Joe said, it's biobanking. And for me to explain to you why we do it, what we do with the samples, why you are incredibly important to us and that we can't do any of our research without you. And so hopefully this will give you a little bit of an overview of everything that we do. So just a little bit of background to us. So the Liverpool Ocular Oncology Research Group, which is where I work, was established in 2006. And this was the foresight of Professor Sarah Coupland and Professor Bertil D'Amato, who many of you may know or have been treated by. I say we're internationally renowned. A lot of people know who we are and what we do. We are very fortunate in Liverpool to be intimately linked with the Liverpool Ocular Oncology Centre, which is currently headed up by Professor Heinrich Heimann, who actually is Sarah Coupland's husband. So it's a good team that's working there. So obviously our focus is rare eye cancers, and in particular uveal melanoma and conjunctival melanoma. So I'm not going to talk about conjunctival melanoma because Yamini did that yesterday, but I'm going to talk about the research that we do on uveal melanoma. And yeah, I've got my little biobank symbol up on the right-hand side because, as I say, that is incredibly important to all the research that we've undertaken over the last 15 years. So a little bit background to me. So I joined in 2008. I came from the Christie Hospital where I didn't work on eye cancer. I've worked on colorectal cancer. I've worked on late effects of childhood cancer, so looking at how cancer treatment affects childhood growth. And then I worked for a short time on breast cancer. So the ocular field when I joined in 2008 was incredibly new to me, but I'm really happy to say that, 15 years later, it's a group that I've really enjoyed working with, and I think the work that we do is really important. So the Liverpool Ocular Oncology Research Group, we're what we call a translational research group. So some scientists, and there's nothing wrong with this, sometimes scientists do the very sort of basic work, so understanding the real intricacies of cell biology. And it's not to say that we don't do that, but ultimately the work that we undertake has to have some sort of relevance to yourselves. So, we want to start with the patient, follow them through their care pathway, be able to look at data, look at samples, undertake the research, and then ultimately feed that back into either clinical trials or changes in practice for yourselves. So in terms of the biobank, so within the clinical care pathway in Liverpool, I also have two other roles. So I'm actually the biobank manager. I supervise all the students in the group, but I also undertake the genetic testing. So for any patients that come through, I have a technician who I supervise who will do the genetic testing on your samples. So I link very closely with the Liverpool Ocular Oncology Centre, and so when the patients attend, and I don't know whether any of you have actually been approached by the nurses to actually take part in our Ocular Oncology Biobank, you will be given a patient information sheet, and then you will be spoken about our biobank, hopefully, by the research nurses, and then you'll be asked whether you would like to consent to take part in our biobank. And the consent form, I have to apologise, is quite onerous. This is due to legislation. We have to ask you things like, are you happy that your DNA is analysed by us? Are you happy that we share your samples and your data with external institutions? So you have the opportunity on the consent form to either agree to those statements or say, I don't want this particular thing to happen to my samples or my data. And obviously that's really important. That's all part of the informed consent process. And so following that, the samples will come through the Liverpool Ocular Oncology Centre up to the pathology department. Because Sarah Coupland is the lead ophthalmic pathologist, then we have a process in place where we can actually access your samples once your diagnosis and all the other clinical aspects have been undertaken. And then I link very closely as well with Joe Sacco, who's the oncologist at the Clatterbridge Cancer Centre. And then also what's happened recently is that the hospitals in Liverpool have merged, and this has been incredibly useful for us because it now means that rather than the liver surgeons being at Aintree, they've actually moved across to the new hospital site at the Royal. And so we link very closely with the liver surgeons there. And so we try and follow as many patients as possible from the start of their pathway through to, if they're high risk and then develop metastases, to understanding the disease as we go through it. So the Liverpool Ocular Oncology Biobank, it was ethically approved in 2011. It's what we call a research tissue bank. I'm the custodian, Sarah's the chief investigator, and we're sponsored by the University of Liverpool. So as I said, we've got a huge number of regulations that we have to follow. There's the Human Tissue Authority, and this was brought in predominantly in 2006 because of things like the Alder Hay scandal that some of you may have heard of, and other tissue scandals, to make sure that everything that we do is regulated, that you know what we're doing with your data and your tissue, that you're happy that whatever you've consented to, you understand what's happening. And so the Human Tissue Authority has actually come into the university this year, so we've been inspected as part of the wider university inspection. And then we also have regulations from the Health Research Authority as well. So every 5 years, I have to go back and renew my ethics. And so the next renewal will be in 2026. And so I'm going to talk to you about some things that's going to change in our biobank before 2026, hopefully. And I think as part of that, I really would like to encourage, again, any patients that want to be involved in a sort of central governance committee of this type of thing. We'd be looking for involvement for any of you that are interested. So yeah, this is our patient information sheet. I've already mentioned about informed consent. And so what are your ocular biosamples? So obviously you have health data that's associated with these that we also try and collect. But these are samples that are taken during your routine procedures. So we're not asking you to have any other additional procedures. These are just the samples that would be normally taken when you come into the oncology centres. So if the sample's then taken, if there's anything left over after the pathologists, the ocular oncologists have actually looked at your disease, then we're allowed to take that. Sorry. I think that was me actually touching my chest. So the samples can be stored either in paraffin blocks, and so I was talking to Scott yesterday, and he said, what is a pathologist? And so a pathologist is someone who's really important in terms of your diagnosis. So you will see Prof. Hyman or someone at Moorfields, but actually everything that they're telling you comes from the pathologist. And so when your samples come up to pathology, they will be put in what we call a paraffin block. And this is it's as simple as it's just a wax block. And what it does is it holds your tissue and allows us to do certain things to it that can tell us things about the disease. Alternatively, we might freeze the samples, and we do this in liquid nitrogen, which is incredibly cold, and then we place them in really low temperature freezers, and this allows us to store them for long periods and then go back to them down the line. Or we might use your samples fresh. And we do this with things like tissue, where we can actually grow patient cells, use them in models that we've got, try and understand how they're behaving, test drugs on them, and things like that. If anyone's got any questions while I go through, because probably lots of aspects of this might be more complex. So if I say anything that's not understandable, just let me know. Yeah, you don't really need to know this. So yeah, we've been in existence till 2011. We've got a huge number of samples in the biobank. This might sound great, but obviously the regulators don't like this, and you don't like this. You want us to be using your samples. And so we are using your samples, and this is just to tell you some of the things that we've been doing with them. So we worked, the top on the genome-wide study was a group in Germany who contacted us, and we provided them with hundreds of samples of blood, and they were looking at genes that may have an association of risk of developing uveal melanoma. And so this was one of the studies that we were involved in. We have also been involved, so the proteomics study was with a group in the USA, someone that we know really well. And so they've been looking into biomarkers that might predict certain aspects of the disease. One of the things that we've done within our own research group, and again, I think yesterday you heard about BAP1. So BAP1 is this gene that changes and is associated with an increased risk of developing metastatic disease to the liver. And so a lot of centres don't have the capacity like ourselves to perform genetic tests. And so what we'd done is we'd used a way of detecting so your gene then gives rise to a protein, and so we developed a way to detect the protein in the bits of tissue that come up to your pathology lab. And so we started this in 2014. This publication was in 2018. And now this is routinely done in the clinic in Liverpool. So when Sarah gets a sample from a patient, then we look at expression of BAP1 protein in these samples. So I don't know whether you can see, how can I walk with this? On the left-hand side here, you've got what looks like brown staining. This means that the tumour is expressing BAP1 protein, and this means it's intact. If your tumour has a mutation in BAP1, then you lose expression of the protein, and this is an alternative way of us determining whether the tumour has a BAP1 mutation or not, just by looking at a coloured marker in the tissue. And I don't know whether you can see these at the bottom, but these are survival curves. So we do genetic testing, and if you have loss of one copy of chromosome 3 or monosomy 3, then the patients are at an increased risk of metastasis. And this shows the survival curve here for patients with loss of one copy of chromosome 3. And this is the nuclear BAP1 negative, so the ones that I showed you on the right. And again, the curve is looking very similar. So it was a way of us showing that actually what we were doing in the pathology lab was actually replicating what we thought based on the genetics of the patient. So like I say, this is now in routine clinical practice in Liverpool, and I know that a lot of other centres use it too. One of the other things that you heard about yesterday was LUMPO. So again, this is another sort of impact, if you like, that we've had from the biobank. So the data that we collated was used to generate the Liverpool Uveal Melanoma prognosticator online. This was done in collaboration with biostatisticians at the hospital. So they've developed this model where you can put in the clinical features of the tumour, the histopathological features of the tumour, and the genetics if it's available. You don't need the genetics to actually run this model. And then it generates an individual personalised survival curve, which indicates your risk over time of developing metastatic disease. And so again, this is something that we introduced. We've got, so this was available outside the UK to use this particular model, but because of medical devices regulations now, it's currently only being used in the UK. That's my understanding. And we're also going to be needing to CE mark this as well. So it's something that we're working on at the moment, because that's incredibly complex and incredibly expensive, which is difficult. And so this has been validated externally as well by groups both in the States and across Europe. So one of the other things, the last one I'm going to talk about that we've been involved in is, I don't know whether anyone's heard of the Cancer Genome Atlas. No, so it was a little bit like, I don't know whether you remember the government introducing 100,000 genomes here in the UK. So this is what the States did before we did it, and they did it much better, in my opinion. And so we contributed, they managed to analyse 80 primary eye uveal melanomas. We contributed about 26 to 30 of those, and it was probably the biggest ever study looking at these tumours in detail from a genetic, protein point of view. And so, again, we've got things like BAP1. I think you heard about SF3B1 and other mutations that I think Heather Shaw and Paul Nathan were talking about with clinical trials. So what's going to change with our biobank? Well, we've realised that probably one other really important type of data that we're not collecting is from bioimages. And so by that I mean images that get taken when you first attend the Ocular Oncology Centre, so the fundoscopy images, the OCT, liver images, so the ultrasound, the MRIs, and we can get really important information from these. You've probably all heard of AI, artificial intelligence, deep learning. Everyone's doing it. It's really important that we are there as well in that field, and that we can use the information that is in these images to try and understand various questions. And so I suppose the other images that we want to collect are the pathology slide images. So I showed you the paraffin wax block. Sections get taken of your tissue from that paraffin wax block, and then we can look at all sorts of things. So you can see an image of the eye and the tumour that was growing in the eye here. We can look at what we call non-tumour components. So the tumour cells sit in like what we call a matrix, a jelly, that influences their behaviour as well. And we can look at protein markers. So they're the stains like the BAP1, the brown stain that I showed you. And that one there is actually marking macrophages, which is a type of immune cell in your tissue. So this is where iCanAid, the fancy name, comes into, but essentially it's us expanding our biobank. And we weren't able to do that on their own. We've had to join with a company that was originally a spin-out company from Liverpool called AIMS. It's now owned by the Arrow Group. They actually house all the NHS data in warehouses in Wavertree. Who knew? You know, I had no clue that like data, you know, from patients was housed there. So they know how to do things. They know how to extract things from the NHS, which we have no clue about. And so we're working with them to generate an infrastructure where we can extract imaging data, any other data that we think is important, and at the point of extraction, and this is really important for the hospital, they get completely anonymized. So what I probably should have said as well, with all the research that we do, no one ever sees any patient identifiers. We code everything with a unique biobank code, that everything's stripped from our data, and so any researchers will not see any patient identifiable information. But obviously that's really important for the hospital, because if we're extracting images from their systems, they want to be reassured that we are anonymizing at the point of extraction. So just to give you a little overview, so this is Max Jackson. He's a PhD student within our group. He's funded by Northwest Cancer Research. So one of the things I should also say, we heard how difficult it is because obviously this is a rare disease. It's very difficult as well to obtain funding in this area. It's just incredibly difficult to get research funding. So Max Jackson, a PhD student funded by Northwest Cancer Research, he's a physics student. We've not had a physics student in our group before. It's really interesting. So he's looking at the AI side of things. So he's taking the fundoscopy, the OCT images, and he's asking questions like, is it a nevus or a melanoma? So things that the clinicians perhaps, find a little tricky, although they wouldn't admit it. Will it metastasize? So whether we can look at the digital pathology slides and not have to do all the stains that we do, because sometimes the samples are really small, sometimes we can't get everything out of them that we need. Can we determine metastatic risk just by looking at your pathology slide with a simple stain? And then can we detect metastasis earlier? So we saw on Paul Nathan's slide yesterday the tiny lesions that the radiologists then see on the MRIs. Can AI detect that earlier? Can we be able to pick up the lesions sooner? And so this is a slide actually that Sarah gave me, and I had to look this up. So this is an article on the BBC website. I think it's in all sorts of newspapers as well. And it was for breast cancer, for mammograms, where they trained an AI tool on millions and millions of images, mammogram images from breast cancer patients. And then they put it into real clinical practice. And there was, I think, 11 patients where they detected an abnormality that hadn't been picked up by the radiologist. And one of those women then went on to have the small lesion removed, and obviously that increased her chances of not having comprehensive chemotherapy, radiotherapy. So it's whether we can do the same with the liver MRI images as being done in other cancers. So I should also say that the bioimaging resource that we're setting up at the moment is just from the Liverpool trusts. Again, I can't tell you how difficult it is to try and combine these sort of abilities with the other centres. So it's not that Moorfields or Sheffield don't want to do this, it's just incredibly difficult to combine the trusts and actually get this information through the regulatory bodies and everything else. But it is something that we're working on, it is something that we want to do. The more data we have, the more accurate this will be. That was just to introduce Yamini. You heard about Yamini yesterday, who's working on conjunctival melanoma. So the next few slides are going to be really sciencey, but I'm not going to hopefully say anything too sciencey. So what are we doing in our research group at the moment with our bioresource and our tissues and the data from patients? So at the moment, we're working mainly on metastatic uveal melanoma, so uveal melanoma that has spread to the liver. And again, I think we heard this yesterday, and I think that this is because, historically, liver metastases have been treated with drugs that were used in cutaneous melanoma, and we heard how different uveal melanoma is from cutaneous melanoma yesterday. So this was, again, I think it was Paul Nathan. These are the mutations that you find in uveal melanoma. Cutaneous melanoma is so much more complicated, so many more mutations. And then this is what we look at as scientists, differentially expressed genes. So basically the genes in red are the genes that are upregulated, the genes in blue are the ones that are downregulated. If uveal melanoma was the same as cutaneous melanoma, then the red and the red would be there, the blue and the blue would be there, but you can see that they're completely opposite. And I think, as patients, that's just been incredibly frustrating that, drugs have just been used willy-nilly with no real scientific rationale for that. And so what we're looking at is the various aspects of the tumor, what we call the tumor microenvironment. And this is it's just for us to understand that in the past, everyone just looked at the tumour cells, but we know now that that's just not appropriate. We know that the tumour cells are influenced by the immune cells. They're influenced by what we call stromal cells, so other cells that sort of sit around the tumour cells, that they're influenced by this extracellular matrix, this sort of jelly that they sit in. And so we're trying to look at all aspects of that. So this is one way that we can look at the immune cells. So we have these fancy techniques now where we can take a bit of tissue, and in the past you used to have to mulch up the tissue and just look at it as a whole thing. Whereas now you can take your section of tissue where you can see all the different cell types in the tissue, and you can go, what does that cell express? What does that cell express? So you're not getting a global overview of the tissue like we used to in the past, where we'd go, oh, well, maybe that is the immune cell that's expressing those. We can actually do it and actually see where these things are in the tissue and actually make sense of them. So this is something that we're doing on the immune cells that are present in the tumours. I didn't put any images up here. I'm actually disappointed that I didn't. So I look at the pathology slides of the liver metastases, and oh my God, how different they are is unbelievable, both in terms of the immune cells that are inside them, the tumour cells that are escaping from the main bulk of the tumour. It's absolutely fascinating, but it shows how we really need to be careful about the sort of questions that we're asking. We need to get these right. It's not one size fits all. We need to be understanding this in a lot more detail. This is Josh, another PhD student. So I was saying to Scott, we've in the past, we've been an all-girl team, not because we wanted to be, it just happened that way. And then last year, we got 3 PhD students, and they were, I'm going to say, boys, males, and the dynamic in the group changed dramatically. It was really interesting, and it's been really good, and they are really focused. I'm really pleased to say that, these students are incredible, 1 year into their PhD, and they're really good. And I think what we've done from the outset is that we've encouraged them to, attend some of the clinical sessions, go and see what happens in the pathology labs, so that they understand what happens to a patient as they go through their journey. So they're not just sitting in a lab going, oh, this is nice, I can, do some cell biology or whatever. So Josh is looking at the matrix, so he's looking at this jelly, and the way he's doing it is that we have a technique where we can make all the cells disappear, and then we're just left with this lattice that the cells sit in. So he's doing various aspects of that. I'm just going to flick through these because there's way too much science in these. And then Karen, she's actually another sort of senior scientist within the group. She's trying to develop models. So what we don't have in uveal melanoma is a very good animal model. I'm not an advocate of animal models. I've never worked with animal models in my career. But what we want to do is we want to be able to not model everything, because I think that's unrealistic, but model certain aspects of the metastatic liver disease. So what Karen's got here is tumour cells in the centre here. So they're marked with, a stain that stains them in brown. And on the outside, we've got what we call hepatic stellate cells. And we know that we have tumours in the liver that actually look like this. So tumours where there's a nodule of liver disease, but actually they're surrounded by hepatic stellate cells that lay down a really fibrotic wall, if you like, walling the tumour off. Now, you could say that's walling the tumour off so that they can't escape, or you could say that's walling the tumour off so that the immune cells can't get in and attack it. So Karen's working on various aspects of that. And this is just a nice little video of what we can do. So we put the uveal melanoma cells in with these hepatic stellate cells, and these are the cells on their own, and these are the cells with these hepatic stellate cells, and you can see they're very different. So again, if we don't use the right models, we don't ask the right questions, we're not going to find the right answers. And yeah, I'll just leave it there and say thank you for listening. Thank you.